Hello, everyone. Thank you for coming today. My name is Erika De Benedetti, and I will manage this webinar. Before to get started, I would like to give you a couple of information. Firstly, this webinar is recorded, and the recording will be available on biostimulant.com and on our social media in the next days. Secondly, I invite you to use the Q&A box at the bottom of your screen to submit questions. We will collect your questions and answer to at least some of them at the end of the presentation. The topic of today is biostimulants in microgreen production. It's my pleasure to introduce our speaker, Christophe Hanachel, postdoctoral researcher at the University of Naples, Federico II. So welcome, and the floor is yours.
Okay, thank you, Erika. Good afternoon, everyone. So as Erika already told, today I will be talking about microgreens production and especially the use of biostimulants. Microgreens is a topic that we are working on for since 2016 with Professor Rufael that introduced us to this interesting topic. And so the webinar of today will be about a small introduction for everyone who's not really familiar with microgreens. We'll talk about the production technologies used for microgreens. I'll talk about a little bit about the market. We'll talk about the pre-harvest factors that can define a lot of qualities in microgreens. And then we will talk about the potential use of biostimulants, a bit about coating the seeds and about the application of the biostimulants in the nutrient solution. So to give a little glance about microgreens, microgreens are defined as young and tender edible seedlings produced from the seeds of various species of vegetables, herbaceous crops, aromatic herbs, and wild plants. And based on the species that we use, they can be harvested from 7 to 21 days after germination. But this range is a little bit approximate because it depends not only about the species, but it depends on which stage we decide to harvest microgreens, since there's no specific definition about the right time to do it. So as we know, microgreens, and you can see in the picture, they are harvested by cutting the seedling at the base. So we can get plants about 3 to 9 centimeters high. And the edible portion is represented by the entire part that we cut, which is the stem, the cotyledonary leaves, and often the true leaves, the first or even the second leaves. But we can never exceed the stage of 2 true leaves. Otherwise, it will not be considered microgreens anymore. In some cases, when they are small and tender, even the seed teguments can remain attached on the microgreens and can become a part which is edible. In the photo that you can see below, these are the seeds of spinach, for example, but since they are not tender, so they are not really appreciated to be found in our plates when we are consuming spinach microgreens. So microgreens got a lot of new definitions. They are also called vegetable confetti. They are called also microherbs, and they provide a very wide range of intense flavors and bright colors. So they don't only give great color to our plates, but also they do change the taste and somehow, and it depends on the species that we are using. So they are ingredients able to enhance and garnish drinks, salads, appetizers, soups, sandwiches, and desserts, as we're going to see in this picture. So they are used on cakes, on a lot of types of food that we can imagine, and also on salty dishes, because microgreen began appearing on the menus of chefs in San Francisco, California, in the United States in the early 80s. And they have been grown in Southern California since the mid-90s. So microgreens are a new category of vegetables that are characteristic with distinct from the they are really characteristic if we compare them to sprouts, that they are more common, I guess, among you. You've heard of sprouts more than microgreens, maybe. And they are also different from the common fresh cut leafy vegetables known as baby leaves. So compared to sprouts, the sprouts consist of the sprouts and the rootlets, and they are generally produced in the dark and soaked with water. And the production cycle consists of a very few days, while microgreens are grown in greenhouses, in open air, or in growth chamber, using soil or alternative substrate, or in hydroponics. And the most important things, in the presence of light. It could be sunlight in case of greenhouses, or it could be artificial light provided by by lamps that we, or LED lamps, HPS lamps, any type of lamps that are adopted for the growth chamber. So microgreens have a longer growing cycle compared to sprouts, and the edible portion, as I said before, is the only aerial part that is deprived from the roots. And which what is different from sprouts, that microgreens you can choose either using only water to grow them, or also use fertilizers, while sprouts we use only water to soak them. And what makes them also different from the baby leaves, that the only edible part is the true leaves. They are also cut, but we take all the above part, and that's what makes them also different. The also different harvest method for microgreens, in case we can decide not to harvest them and sell them alive in their trays, so they can have a longer shelf life if placed either in the supermarkets, either if they are in the kitchen of the chefs, because in this case, they can cut it and use it straight away. So the terms microgreens and baby leaf do not have legal definition, but are used to describe two particular categories of the products. On the other hand, sprouts are legally defined, and their production and marketing must comply with strict rules, as the risk of microbial contamination is higher than that of microgreens and baby leaves, as I already mentioned, because it's soaked in water and in dark. So there's a big probability of contamination in case not all the phytosanitary rules are respected. And as you can see in the picture, from the same seeds, I can have sprouts, microgreens in the middle, and baby leaves in the last pic. So microgreens are important, especially because they play a role in agrobiodiversity. They can help or contribute as well to the preservation and valorization of many local varieties that are at risk of genetic erosion or extinction, sorry, because some local varieties, they get forgotten over time, because we always tend to go for all the new kind of seeds, and we forget what nature offers us in our zone, because in each geographical area, there's a huge resource to explore, since every area has its own plants and species. So, and usually when we go to choose the selected or improved varieties of vegetables, and we take these seeds to use it as microgreens, these seeds of vegetables, usually when they go through breeding, in case to have better yield or better resistance, they lose a lot of the phytonutrient content characteristics. So the plants do not offer what the wild varieties or the spontaneous varieties can offer us. And in addition, the modern consumers really are searching for food that can help boost their health. So they search to eat also microgreens. So for the production of microgreens, high quantities of seeds of good germination and low cost are required, especially because it's the higher cost in this production. And the seeds of local varieties often have these characteristics. In addition, microgreens enter in the food security. Why? Because in an agricultural system, when we have the diversification, it means that we have a pillar of sustainable development. So the consumption of the community is not based on one kind of production. So we have different kind of production in case of any problem. There's something to relate, something to sustain the other lack. So as well as in farms, we can grow microgreens in urban gardens. We can grow it at home and on the balcony, on the window. So it's very easy to grow it. We don't need to say that it's something done by expert. It's also something very easy to do it at home if you have the basic instruments and the basic knowledge. In addition, microgreens, talking about 0 kilometer microgreens, is something very interesting, which could be provided by the urban agriculture. 0 kilometer is a term that started in Italy, where you eat and where you produce and eat your food in the same area without even needing a lot of transport. So you don't contribute to a kind of pollution directly or indirectly. So the food is present in the same area. And also in case of any interruption of the supply chain, having 0 kilometer microgreens in the cities is really important. In addition, having microgreens could offer an accessibility of food to the poorest populations in the world. And here I'm talking about the qualitative point of view, because in case you can get, you have access to seeds from your area, which they cost very low, you can plant it and get microgreens, and then you can get a meal very rich in phytonutrients that can help the poor population also get what they need of their daily nutrients. In addition, microgreens are eaten raw and intact. So in this case, there's less food discard and losses, and there's no degradation of phytonutrients, especially for the food that goes through cooking. So that's why it's important that it is eaten raw. So in order to choose the species that we want to grow, speaking of an agronomical point of view, we have to check the quality of the seed. So the seeds have to be homogeneous and they have high germination, because when we grow microgreens, we aim to harvest all the seedlings at the same phenological stage. So we cannot afford to have seeds germinating in 2-3 days, having different time of germination, because when we go to harvest, and then we will not have all the seedlings in the same phenological stage. And why we need high germination? Because also we cannot put a lot of seeds and that they will stay on the level of the substrate and they will not germinate. So during the growing cycle, these seeds that did not germinate could create phytosanitary problems. So therefore, it's better to have most of the seeds germinating. In addition, they don't have to be chemically treated with any fungicides, for example, because we're not sure if, in case the cycle is very short, that the chemicals will be degraded and we will not offer any problems for the consumer. In addition, they should be hygienically safe as well. No contamination of the seeds, which could be very common. And in addition, as I said before, since it's one of the most expensive cost of the production, this price should be very low. In addition, we have to choose seeds. They don't have any thermal or environmental requirements that differ or is very hard to execute. In addition, when we choose our species, there's several aspects to take care of. So it depends if it's a product sold to a restaurant or a chef. Maybe he's interested in the shape of the microgreens and the color of microgreens, because the microgreen species can offer us a myriad of colors. So depending on the species, we can go from green, we can go to dark green, to violet, to red. We can even have the stem that has different color. For example, Swiss chard, there's varieties with stems that are yellow and the leaves are green. Or amaranth, where we can find that it's entirely pink. So in addition, we have to choose the texture also as well. What are we searching to add to our plate that we are preparing, or the flavor, for example? Like, let's say, for example, borage has a taste of vanilla and it's a little bit sweet, so it could be a good addition to the dessert that we are preparing. In addition, there's a lot of species that we can use for microgreens growth. And a lot of botanical families are really known for microgreens, and especially we have to talk about Brassica family, which we have cauliflower, Chinese broccoli, cabbage. So there's a lot of species in Brassica that they are really famous for the cultivation of microgreens, and also the new species that are really now used, like Mizuna, Komatsuna, Pak Choi, which they can offer also really good colors and taste. In addition, we have also other families that are less familiar with microgreens, but they offer a big potential. And we can talk about also carrot, celery, parsley, coriander, if you are courageous to taste some strong microgreens, and anise as well. You will be surprised, but anise as well could be grown for microgreens, cumin, and in addition, we talk about basil, that is important. Also, cucumber can be used, purslane, jute, borage, lettuce. We can use amaranth, as I already said, spinach, quinoa. We can use garlic, onion. And in addition, we have a lot of also diverse family that we can use. We can use herbaceous plants like oat, wheat. We can do also some microgreens with leguminous plants, chickpeas. Who I'm sure that one of the audience has already grown some chickpeas or some wheat before Christmas under his Christmas tree, and we never knew that it could be edible. So now I think we can encourage people that in addition to decorating, you can also use it to eat and have a better health style. But we have to be careful because some species cannot be eaten. So we have to carefully evaluate the edibility of each species at the seedling stage. And we have to exclude spontaneous or domesticated species whose seedlings are not edible. For example, species of vegetable belonging to the Solanaceae family, like tomato, pepper, and eggplant, which should we completely avoid planting and consuming as seedlings, since they contain high levels of solanine, which is not good for human health. So what are the nutritional properties of microgreens? So intended for increasingly demanding and above health conscious consumers. As I said, consumers nowadays are really health conscious, and they are really looking towards good diets that can boost their immune system and their health overall. So microgreens offer an excellent nutritional profile. Therefore, they are called functional foods or superfood as well, because they provide bioactive compounds like vitamins, carotenoids, phenols. So all the secondary metabolism that we can find in plants that are really useful for human health. They are as well a good source of potassium, calcium, and microelements. In addition, as I said before, the raw consumption of microgreens has the advantage of avoiding nutrient loss or degradation of vitamins. So how to grow my microgreens? So we have to select the growing system. When we talk about commercial level of production, it has to take place in a protected environment. So it could be a greenhouse, it could be a growth chamber, or also it could be in vertical farming. So we have to choose in which place to grow it. On a commercial level as well, the production takes place in containers that could be plastic or biodegradable materials. We can see here like big tray. If we go for large production to sell, we can also go for small trays if we're doing it in home. So it depends. So what's the aim in order to choose the tray? It could be done on channels or benches, as we're going to see in the next picture, or also could be grown in floating panels. So these are the channels, the bench, and these are the channels where we can also grow microgreens. So how to choose the substrate when we grow microgreen? There is a critical aspect for several reasons, because the substrate can have a big role in productivity, because it can increase the growth or it can block a little bit the growth. So for example, peat moss, we're going to see an example later on about how peat moss based substrate can increase the growth. The quality is influenced because the substrate can influence, as we said, the growth as well as the dry matter content. Sometimes it could create some stress, and it could increase the accumulation of secondary metabolites, which can be a positive increase in the quality of this product. As well, the The substrate has a big influence on the growth cycle, on the duration of microgreens. When it's speed-based, it's among the shortest cycles. When you change the substrate, it could be fibers or a capillary mat or anything else, the cycle could be prolonged by 2 or 3 days, depending on which phenological stage we have chosen to harvest our product. In addition, there's also the sustainability of the production. It depends. If it's a big company, they have to carefully examine what substrate they should use, if they are creating any pollution, if they have the accessibility to buy this substrate, if they can found it all year long or not. So there's a lot of factors to consider. So the substrate for a good germination should have a good porosity, they have a good water retention capacity, and good level of root system aeration. It has a pH range between 5.5, 6.5, and a relatively low electrical conductivity, and definitely it should be free of heavy metals and pollutants. We don't want to produce microgreens with some residues of heavy metals or other, and it should not be microbiologically contaminated in order to avoid any phytosanitary also problems for the future, and also it could be sterilized. So to have an idea about the cultivation substrate that we can use, it could start for peat or peat-based substrate, soil if it's in a hobby level. We can use other substrate like perlite and vermiculite that we can mix it with peat. We can use coconut coir, agave fiber, a lot of natural fiber, and we also use the cellulose sponge. We can use capillary mat, and we can avoid using the substrate and go opt for a floating technique. As we can see here, this is a floating technique. This is capillary mat, and here on the right we can see coconut fiber. Okay, so for growing microgreens, we have to check the quality of the seeds. So because it has a very great influence on the spread and uniformity of germination, as I already explained, and it has the most cost, as I said. So we need high germinability. They should not be chemically treated. They must not contain foreign bodies because we can risk to have it when we harvest, and they should have excellent purity with no weed or inedible species that we don't want to have in our plates. So sometimes it is advisable to wash and disinfect the surface of the seeds, especially if it depends on the shape of the seed. So we can do some physical treatments like heat or high pressure or radiation, or we can do like chemical treatments like sodium or calcium hypochlorite solution, or hydrogen peroxide or ethanol. And then definitely the seed can be washed again and then used for the plantation. So how do we choose the density that we want to go for? So in this work that we did once, you can see that the more the weight is higher, or the shape of the volume is higher, the seed the density, the sowing density is lower. Usually in the literature, they opt between 1 to 4 seeds per square centimeters. So, but we in our studies, we also try to go between 4 and 8, and even until 8, we found very good results. So if you want an example, tatsoi that has a very low weight. Here we assess the 100 seed weight. We noticed that the sowing density is high. It's around 7 seeds per centimeter. So here we expressed it in a square meter. So it's all depends. It's opposite proportional between the weight of the seed and the density. So how to germinate microgreens? So usually you can put the trays one on top of the other after you broadcast your seeds on the top of your substrate, or you can leave it on the surface and put a transparent cover in order to maintain a high humidity. I don't suggest to put any substrate above the seeds, because sometimes the substrate can be stuck on the teguments and on the leaves, and we could stay all the cycle. So I would prefer to keep the seeds on the top. During the germination, the irrigation is used from above as an overhead mist. And after the germination, we started with a sub irrigation. And we use sub-irrigation when we have the formation of the seedling in order to not create any humidity, excess humidity on the shoots, and to limit the occurrence of any phytosanitary problems. And then the fertilization is essential if you want to have a good production. Nonetheless, I already said that it's possible also to produce it with only water, but if we want to have bigger plants, better looking plants, somehow we need to apply a small amount of nutrients. So for a commercial production, let's say, we can go also for have a whole blend solution, but in a half or even quarter strength. So reaching around, if we say about quarter strength nutrient solution, we can be around 400 microsiemens as a electrical conductivity. So in the harvest, and there's some stuff that we should take care, and the post-harvest conservation. We can say that microgreens cultivation cycle, as said before, lasts from 7 to 21 days, roughly. It depends on the genotype, the cultivation system, the climatic condition, and the choice of the harvest stage. It could be done manually with the scissors, or with a blade, or with an electrical blade. And after harvest, we should immediately cool it, because the shelf life could be very short in case the temperature are not respected. And of course, we need always, as we do in leafy vegetables, we have to wash the cutting tools, we have to use gloves, we have to be very careful in order to not have any problem that we take it from cycle to the next one. And alternative to harvesting, as I already said, we can market it in trays as a living seedling, and then the consumer can cut it on the spot. So if we want to talk a little bit about the microgreens market, as I already said, the Brassica family is among the leader in this share of the market, with broccoli among the most consumed one. As we see, there's also a place for peas and basil. And as we see, as for the prediction for the market in the future, other also are expected to show. And if we want to speak about the market by farming, how it is done, we can see that most of the microgreens are produced in vertical farming, then in commercial greenhouses, and the rest part, it's on the level of home, hobby style, or in other types of farming. Who usually to whom we usually grow microgreens, as it's present here, we can see that food service is among the highest consumer of microgreens, because as we said, from now on, it's becoming really a trend to be used in restaurants, or because if I'm sure that when you are visiting restaurants these days, you can see that a lot of our plates are now decorated with small colored plants. And if you wonder what it is definitely microgreens. And then we can see the rest being sold in the supermarkets, but it's not really famous, because you cannot find it in a lot of supermarkets. If we want to talk which are the most region that we can see microgreens, or they are produced and consumed, we can see that the first place is for the North American market, followed by Europe, then by Asia Pacific, and at the end we can see the Latin American area and the Middle East. If we go to search a little bit for the word microgreens, we can see that the research about microgreens didn't start relatively long time ago. So it's around 2004 when we started seeing some scientific publications about microgreens. And in the recent almost 10 years, we can see it started to rise in a big matter, and it's being really a topic that is interesting a lot of scientists. So if we check by the number of publication by institute, we can see the University of Naples, Federico II, is among the leaders in doing publication about this subject. And especially also we see Italy, that in the second place after the United States. So we definitely thank Professor Rufael for introducing this topic, microgreens, and to work on it. Therefore, we find the University of Naples among the pioneers in this topic of research. So there's a lot of factors that can influence the quality of microgreens, and especially the pre-harvest factors. First, we have the selection of the species or the cultivar, because we know that each genetic background can dictate what the plant would be, how much it will grow, how much of secondary metabolites it will accumulate, how much it will offer to the people who's eating. The harvest stage can have also an effect on microgreens, because as I said, there's not a clear definition when to harvest it, because you can harvest it at cotyledon's stage, you can with one true leaf, we can harvest it with two true leaves, so it all would affect the quality. There's the nutrition management that could also influence the quality of microgreens, also the growth, also biofortification. When we add micro elements to the nutrient solution, the growth substrate could influence microgreens. And also when we are working, especially in closed environments like growth chambers or vertical farming, the light has a big influence. So we're going to start with some examples about these pre-harvest factors by mentioning some few works that we did. So I will share with you this first work that we did on microgreens, where we cultivated around 13 species or subspecies of microgreens. It's almost like a screening to get to know a little bit better all the species that we can encounter in this world of microgreens. And as we can see, for example, that the phenolic profile of microgreens is strongly influenced by the genotype. And the variability of the phenolic compounds is closely related to the taxonomic origin. We can see that in the center of the species A, we can see almost all the Brassica species, where we can find coriander that belongs to a different botanical family in a different angle, and as well green basil. So these phenolic compounds are like a very good, like we aim to have a high content of these phenolic compounds, since they can really influence the phytochemical quality of microgreens. So we don't only seek for growth, but only we seek for good content of secondary metabolites. And a different work that also characterized a different pre-harvest factor, we worked on the growth stage. So as we see in the picture in this work, that we did it in a growth chamber, we used 4 species of Brassica. And as we can see in the first, the down below row, the harvest stage was at the cotyledon stage, where we can see the emergence of the first true leaf, where in the second stage that we chose to harvest microgreens was the formation of the first true leaf and the initial of the second true leaf that we can see for all the microgreens. So this we can have a look at the growth stage the day of the harvest of the microgreens. So we have 2 green species and 2 violet species. So as we can check in the results that we obtained, that the yield in the second stage, so with the formation of the first true leaves, definitely engendered a higher yield of all the 4 species. While, for example, if you want to check one of the secondary metabolism compounds, the anthocyanins, we can see that for the green species, we didn't have any change between any significant change for the 2 species, while for the red or violet species, we saw that for Komatsuna, there was an increase of anthocyanins in the second harvest, in the second stage, while for Pak Choi, the first stage showed higher values. And another pre-harvest factor, we did another work concerning the nutrient deprivation that we applied on 3 species. We chose for this experiment 3 species that are known to be high accumulator of nitrate. So we wanted to work to see if this applying some kind of nutrient deprivation, because we always use a quarter strength solution to irrigate our microgreens. So by doing day before the DBH is day before harvesting. So when we applied 0 days of deprivation, the yield was the highest. And the more we applied days of deprivation, the growth or the fresh weight reduced gradually. And talking about nitrate, we saw that without applying any nutrient deprivation, Rocket, for example, had around 2,500 milligram of nitrate per kilogram of fresh weight. And anyhow, it was less than what is allowed for green vegetables. So by doing this deprivation, we saw that the accumulation of nitrate decreased drastically. And the more we went further with the deprivation, the nitrate was even lower and lower. So we see at the end for all the species that by doing this strategy, the nitrate is very low. But then there's a compromise. We have to decide whether we want these nitrate so low and we can risk to reduce the fresh weight. So it's here of the grower and the scope and the aim. What is it? So we can decide which type of deprivation we want to apply. In a different work, also on Brassica species, when we have Brussels sprouts, cabbage, and rockets, we aimed also to do a full cycle, or completely with water, or completely with a nutrient solution, a quarter strength. And we can see that when we applied the nutrient solution, all the yield increased significantly. With the rocket, almost doubling the yield when we fertigated with a quarter strength nutrient solution. As for dry matter, for cabbage, it was steady. As for Brussels sprouts, it reduced, as well as for rocket. And if we want to go for nitrate, we can also see that it was reduced drastically when almost being under 10 milligram per kilogram of fresh weight when we applied only water. Talking about quality, here we can see that with the nutrient solution, we had an increase, a significant increase in total chlorophylls for Brussels sprouts, for cabbage, while for rocket, there was an increase, but it was not significant. Also for total ascorbic acid, we can see that it increased in Brussels sprouts with the application of the nutrient solution, while for cabbage and rocket, it did not, only for Brussels sprouts. Also for total anthocyanin, we can notice that with the nutrient solution, it decreased, especially for cabbage, it's very obvious. Then, if we go to, I will show you another aspect of the pre-harvest factors about the substrate that I already talked about, that it could influence also the growth or the quality. Here in this experiment, also we chose 3 species to test the effect. As we see, we have a peat-based substrate, we have coconut fiber, we have a capillary mat in the center, and we have a cellulose sponge. As we can note in this experiment where we used coriander, kohlrabi, and pak choi, that when we chose the substrate, it had a big influence on the canopy height, and not only that, also on the germination. For example, coriander germinated first on the peat-based substrate, while on the other substrate there was a delay about 1 to 2 days after peat moss. So that also could influence, if it depends on if you want to produce quickly and then have also higher plants and higher yield, and then we can go straight for peat moss based substrate. But then we can have, as we see, less dry matter contact. And if we go to all the fibers or the mats or the cellulose sponge, we can see a difference in the dry matter accumulation, which is way higher than the peat-based substrate. But then we will have less yield when we opt for these alternative substrates. In a different experiment also, talking about the pre-harvest factor, where light is really important, in this experiment on parsley, we did apply 3 types of lights: red, dominance of red, or blue, or RGB, where we have the full spectrum. And in this case, we can see that the blue light did have a big influence. As we saw earlier in the picture, we can see that this short tray of microgreens is grown under blue light, while this higher tray is grown under red light. And we can see when we measured the hypocotyl length that under blue light, it's almost the half of the one grown under red. And also the yield was less than the one grown under red plants. So here we talk about the biostimulant application, because previously we were saying that the scientific work on microgreens are getting higher by the years and really getting the interest of the scientists. But if you go searching the use of biostimulants with microgreens, you can barely find like 2-3 works done on this topic. And our university has one of these works, because we found that why not using biostimulants, because biostimulant has a lot of potential. So why not using also these biostimulants on microgreens and see if it's feasible in this short growing cycle to even get positive results. Because biostimulants in general, and especially protein hydrolysate, they contain signaling peptides and free amino acids that can enhance the seedling growth and as well the vegetable quality, because they are able to trigger some kind of metabolomic pathways that can increase positively the quality of the vegetable. The protein hydrolysates are taken through root absorption in hydroponics, in case the application was in hydroponics, and they can be converted to the needed plant compounds in case, especially if they are from vegetal origins. In addition, they are highly available for plants, unlike in substrate conditions where microbial competition occurs. In addition, protein hydrolysase can increase the root growth due to hormone-like activities. They stimulate nitrogen and carbon metabolism, and they modulate the antioxidant system. In addition, protein hydrolysase change the root architecture from length to lateral root branching to root biomass, and thus they increment the root system surface area and increase the absorption of the nutrients and the nutrient use efficiency eventually. So we decided why not do like kind of seed priming, or as well direct application of biostimulants in the nutrient solution, and see what can we obtain in such cases. So as a first experiment, we did seed priming. So in this experiment also we used brassica seeds for this experiment. You will be wondering why we're always using almost brassica, because I repeat, it's one of the most used species for microgreens, and it's very easy to grow and it's very fast. So in this experiment, we applied for the seeds one hydropriming, like it's a water priming, and we did 2 biopriming using 2 different protein hydrolysates. And then we went to broadcast our seeds. And as you see in the first row, where we see the control plants, we can see that after 1 day of sowing, the control seeds are already a little bit late in comparison to all the other treatments, the biopriming and the 2 biopriming and the hydropriming. And on the right, we can see an advanced stage where we can see that control is a little bit late compared to the primed seeds. So this experiment was as well done in a growth chamber. So what we can notice in this experiment, that there was no difference in fresh weight or hypocotyl length, but we saw a small significant increase in dry weight. In addition, we can see regarding the quality of these microgreens that all the priming procedures did increase the antioxidant activity. By using 3 different methods, we can see clearly that there was an increase in the antioxidant system of the plant. In addition, if you want to go to check nitrate, we can notice that Mibuna have no kind of changes, where as well in Komatsuna that we see very, very little change in the nitrate. So we can say that it doesn't have any effect. We can check the magnesium. I'm sorry for the technical problem, but I cannot see the upper part. We can check that also the magnesium was really influenced by this procedure, where we can see that all the 3 priming did also increase the magnesium, where we, for the calcium content and some of the rest, there was no changes. As for total phenolic acids, which is also related to the antioxidant activity that we saw earlier, we can see that for Mibuna is the green species, the cultivar, we can also find that this type of priming, especially the biopriming with the two kinds of protein hydrolysates, had a big increase. Whereas for Komatsuna, we can see a significant increase, but it was not that clear or strong as it was for the other species. In a different experiment, we wanted to try the protein hydrolysates in a direct contact with the roots. Because we know sometimes when biostimulants are applied through the leaves, a lot of environmental factors can affect the absorption and also the effect of these biostimulants. And sometimes we can also see that there's a lack of response. So that's why we said, why not trying to do this application direct in the root system? And in this published work that we did also under the supervision of Professor Rufael, we were the pioneers of adopting this floating graft system for microgreens. And in addition, we added the protein hydrolysates in the nutrient solution. So in this case, we adopted the two species of, we had carrot and dill. The application of the biostimulant here only increased the yield of dill. As for the dry biomass, it was as well increased only for dill, and for the dry matter content, it decreased only for carrot. As we can see below, the roots in this system, we can see that the protein hydrolysate increased the growth of the roots. We can see as well the effect on color. And we can see that there was an increase for dill, there's an increase in the brightness, and we had also for carrot an increase in the green parameter, which is important because we know that the appearance of vegetables play a crucial role when customers are choosing their vegetables. So when you have an increase in color, that could be a good attribute. If we go to assess the secondary metabolites, we can check that the application of biostimulants did increase the anthocyanins in carrot in a very, very big manner, while it reduced it in dill. While for total ascorbic acid, we saw a decrease for carrot and an increase for dill. So if we want to talk in general, the response of biostimulants are still yet to be understood better because it is species dependent and sometimes also cultivar dependent. If we check also in this experiment on qualitative parameters, we see that the application of biostimulant, the protein hydrolysis in the nutrient solution, increased the soluble proteins and also the essential amino acids were increased in carrot and dill, and also the total amino acids were also increased by the application of the protein hydrolysates, because protein hydrolysates offer like free amino acids in the nutrient solution, and they are readily available for plants to be used for their metabolism. In a different experiment as well, we applied the biostimulant, the protein hydrolysates, in the nutrient solution, but this time we did it in a growth chamber, but because we wanted also to have an experiment that could be also applied for growth chambers and vertical farming. So in this experiment, we had a little bit different trend because the increase in the fresh yield was observed for both species, but in a different measure. Because if we check the increase of press when we applied the protein hydrolysate was around 5%, while when we check the one on kohlrabi, the increase of fresh weight was around 20%. So the biostimulant was significantly active on both species, but in a different matter. And for dry matter, we had a decrease for kohlrabi, and while it was almost steady for cress. And another good aspect of this is we had a longer hypocotyl for the application of the protein hydrolysates, which is also another aspect that we can look at. Because if we have a more vigorous microgreens, it will definitely be more appealing to see it on the plates that we are preparing. In addition, we also did an assessment of the color. We also can see that the application of the protein hydrolysate increased the brightness also, and increased the greenness of the microgreens. As for nitrate, for example, we see that the addition of protein hydrolysate did not influence the accumulation of nitrate. And for the other minerals, we can see an increase of the accumulation of magnesium and sulfur when we applied this protein hydrolysate in the nutrient solution. So as a conclusion, we can see that the pre-harvest factors, they dictate the microgreens qualitative characteristics, because every aspect that we discussed, they could influence either the yield or the quality. So it's very easy to manipulate, although the genetic background plays a big role, but our management and the application of pre-harvest factors can have a very decisive saying in the quality that we are aiming for. So we notice that even if the cycle of microgreens is very short, the application of biostimulant was really able to increase yield and the qualitative aspect, but definitely in a cultivar or species dependent manner. In addition, the biopriming of these seeds that we did definitely showed to be able to modulate the dry weight, and most importantly, the total phenolic acids content and the antioxidant activity of Brassica species. So although the priming did not change the time of the growth or anything, or the weight, the fresh weight, but we were definitely able to boost the qualitative side of the production of microgreens, which is especially a very important manner when we're offering for good quality to consumers. In addition, the direct application in the nutrient solution in the last two experiments that we see was we were able to see an increase in yield and in hypocotyl length, which is, as we said, very important, as it happens for cress and kohlrabi. And we could manipulate as well the qualitative attributes by the addition of these protein hydrolysates. So I want to thank you for your participation in this webinar. I hope everything was clear and beneficial.
Thank you very much for this interesting presentation. And now we will have a Q&A section. I just wanted to remind that it's possible to submit your questions using the Q&A box at the bottom. We have already received some questions, so I will start immediately with the first one. You showed different trials with biostimulants, and referring to the trial with the nutrient solution, the question is, when was the biostimulant applied, and was it applied frequently?
Okay, so yeah, for example, for the experiment that we did in the growth chamber with the two species, cress and kohlrabi, we applied the biostimulants when we turned on the light. Usually we turn on the light when the germination occurred and the seedlings start to emerge. So when we are sure that the cycle will clearly begin under the light conditions, we added the biostimulants. And did we use it frequently? Every time, since in these trays, there's the capacity of around 1 liter, 1.1 liter. So, and in the middle of the growing cycle, the microgreens are growing very fast and they consume a lot of the nutrient solution. So basically on a daily basis, you have to add the nutrient solution that is lacking in order not to have the roots not completely immersed in the nutrient solution. So we were every time adding the nutrient solution, we were adding the fresh biostimulants in order to maintain the same concentration in the nutrient solution.
Okay, thank you. I will continue with another question. The question is, do biostimulants allow to reduce the use of fertilizers in microgreens production?
Okay, so as we said that in microgreens, it's not necessarily to put fertilizers if you're growing on a small scale or for your own consumption. But if you're talking about a large scale production and you need to have a good production to sell, then a small amount of fertilizers should be added. So we don't have a clear study yet on it, but since we know that biostimulants do increase the dimension of the roots, the architecture of the roots, they can, the lateral roots as well, the number of lateral roots are increased. So they will definitely help increasing the nutrient use efficiency and the absorption of nutrient. So it would definitely be useful. But since we, for example, on microgreens, I cannot be very clear, but we have similar studies done on mature basil plants that I can even recommend to read these works, where the addition of biostimulants, the same biostimulant protein hydrolysase from vegetable origin that we used in this experiment was added to basil. And the addition of this biostimulants was making able the lower concentration of the nutrient solution to produce the same as the double concentrated nutrient solution. So I can assure that the use of biostimulant can help us reduce the use of fertilizers in such type of cultivation, especially in floating systems.
Okay, thank you. We have other questions. Someone asked if can you tell us about what are the types of protein hydrolysase you used in the experiments? And I will add also another question. If have you tried other types of biostimulants like seaweeds or microorganisms?
Okay, so the two types of hydrolysase that we used are from vegetal origin, as I said, and they are from leguminous plants. One of them, if you go, I can send you the paper where you can see exactly what is the product used. So I definitely say again, it's from vegetal origin, leguminous. But for the other kind of biostimulants, we haven't used it yet, but since it's a new topic and we are willing to use all kinds of biostimulants, we're definitely keep in mind to use seaweeds or other type of biostimulants like microorganisms, because I think we should still discover a lot in this kind of applications.
Okay, thank you. I will close with the last question. So thank you for your presentation. And the question is the plausible mode of action of biostimulants on microgreen will differ from crop plants, or you think based on your experience that it will be the same?
Well, I will definitely say it is the same, because when you do, for example, as I said already, that having the same application, like in a floating system in water, it could have the same high microgreens or mature plants. But if you go to spray it in the field, for example, you do a foliar application, then I cannot confirm that we can have the same results, but because in the field, for example, we can have a lot of environmental factors that can influence. But in general, we can affirm that biostimulants have a clear action, so it will be definitely be beneficial for the plants. If it's not beneficial on the yield point of view, it will definitely be good on the qualitative point of view, or even in the stress tolerance for the plants. So the application of biostimulants is always beneficial for a good sustainable agriculture.
Thank you. Thank you again for your answers. This was the last question for today. We have finished our time. I would like to thank you again for the nice presentation of today. And I would like, thank you. I would like also to thank everybody for attending the webinar. And if you still have questions, you can submit them on biostimulant.com website, where there is a specific section called Ask the Experts, and our scientific committee will get back to you with the answers. I would like to remind you that the recorded version of this webinar will be available soon on biostimulant.com and on our social media, so stay tuned. Have a nice day, everybody, and I hope to see you on the next webinar. Bye.
Bye-bye.